Okay, so it's great and my pleasure to continue the Melastome virtual seminar series. Um, we have today Lucas Majure. Lucas is currently at well, I don't know if it's officially curator, director of the herbarium at the
curator, yeah, whatever you want to call it, yeah.
At the University of Florida Natural History Museum and a professor at the University of Florida. Lucas started his life botanizing in the middle of Mississippi with lots of melastomes. Well, maybe not so many.
A handful, yeah.
A handful. Then he went to Florida where he worked on Opuntia and polyploidy. And because that was too easy, he decided to start working on melastomes. He did a postdoc with Walter Judd, and then went to the Desert Botanical Garden, and he couldn't stay away from Florida and came back a few years ago now. And while he's still working on Cactaceae in the Caribbean, he's also continuing his work on melastomes. But that's only, I think, his side projects, because he also works on P. Cramniaceae and Poaceae, biogeography of the Caribbean, biogeography of plants in the Southeast US. So, you know, quite a few different interests in Lucas's life. So at least this presentation today joins two of his interests, which are the melastomes and the Caribbean. It's my pleasure to have you here, and take it away.
Great, thanks so much, Fabian, for the introduction. It's very wonderful to be with you guys today, and I'm happy to share a little bit of work that we've been doing in the Caribbean on melastomes. It's sort of with the focus on Miconia. So let me just, So essentially what I want to do today is just sort of talk about sort of like going from the broad picture to sort of a narrowed focus of what's going on with certain groups of melastomes within the Caribbean region, and specifically talking about this really large clade, Miconia or tribe Miconii. A relatively large clade of, close to 2,000 species, something like that in the neotropic. So not insignificant, right? Again, thanks. It's great to be here with you guys. I want to start out with just sort of talking about how I'll focus the talk in terms of, when I say Miconia, what exactly am I talking about? Because it's not unconfusing, or it's one of these situations that, we've had to deal with over the past number of years. This figure from a recent paper of Fabian's from the book that was just recently published kind of gives you an idea of how complex the situation is across the tribe. And so essentially what this figure shows is what was traditionally recognized as Miconia in yellow, and then everything else in different colors are essentially other genera that have been recognized within this tribe, right? And so you can see that there were huge problems with non-monophyly across the tribe Miconii. And so that's why everything that I'll be talking about today, I'll be calling it Miconia in the broad sense, Miconia cinculatum. And so if we take a look at some of the Miconias that occur across the Greater and Lesser Antilles, for instance, in the photographic plate on the right-hand side of the screen, you can see that we're looking at things that oftentimes, it's kind of hard to wrap your brain around, that we're calling these things all of the same genus, right? Because we have an incredible diversity of inflorescence types, of vegetative morphology. We have plants that essentially crawl along the ground. We have trees, that are 10 meters tall. They essentially do it all in the Caribbean. We have inflorescences that are incredibly large and expanded with dozens, if not hundreds of flowers. And then we have inflorescences that might be reduced to just one flower within this group. And so it's an incredibly diverse group from a morphological standpoint. They also grow in a wide diversity of habitats, which I'll get into a little bit more. And so when you look at Miconia, in the Caribbean region, sometimes, they're head scratchers because there's such a broad diversity of morphology. Of course, if you look at this group sort of in the broad sense across its range, you really see the same amount of morphological diversity. So I guess it shouldn't be too terribly surprising. So when I just sort of want to focus us and get us in line with where we'll be sort of situated for the rest of the talk in terms of geography. So I'm going to be really focusing on the Greater Antilles and Lesser Antilles, but mostly on the Greater Antilles today. And so to focus us geographically, for those that might not be, fully familiar with this region, of course, we have the Bahamas here that I don't have labeled. Then we have the Greater Antilles consisting of Cuba, Jamaica, Hispaniola, Puerto Rico. And then, of course, we have the Lesser Antillean islands, this arc that extends between Northern South America and the Greater Antilles. A lot of my focus today will be on the Greater Antilles because, for instance, Cuba and Hispaniola really have some of the highest numbers of species and overall species diversity within Melastomataceae and specifically Miconia within this group in the Greater Antilles. So why the Greater Antilles in the first place? Or I should say, why the Antilles in the first place? Why the Caribbean region? This is one of the top 5 areas in terms of biodiversity on the planet. It's a biodiversity hotspot. So it's a very small area in general because we're dealing with insular systems here, but there's an incredible amount of endemism within this area. So of the roughly 12,000 or so species that occur throughout the islands, and upwards of half of those are endemics to the area. And so it's a really fascinating place to work and a really fascinating area to try to wrap your head around how that diversity has actually evolved. So to do that, we kind of have to think about the geological history obviously has played a super important role in not only deriving the habitats and ecosystems that we see today, but also given the soil types and sort of those geological processes have actually led potentially to why we see such a diversity in the islands. So I have some relatively confusing figures here on the left from Mann, who published a really great paper in 1991 that discussed mountain building, especially on the island of Hispaniola, but essentially gave a really nice overview of the overall process, at least processes, the way we think they have unfolded, specifically with regards to the Greater Antilles. And essentially, this has been, a geological process over millions and millions of years, but essentially by sort of, mid to late Eocene, the Caribbean islands were essentially where we, see them today. But they were not necessarily in that sort of conformation that we see. So if we think of the island of Cuba or the island of Hispaniola, those islands are actually composite islands derived from multiple land masses, Cuba being derived from at least 3 different land masses, Hispaniola being derived from essentially 2 major land masses. And those land masses, specifically with regards to the island of Hispaniola and Cuba, essentially became these, large land masses that we see today in roughly mid-Miocene to sort of latter in the Miocene times. And so we're, between 9 and 15 million years ago. And with the accretion of different land masses, we also had significant mountain building. And so the mountains that we see today are sort of the products of these plate tectonics. So where you had the Caribbean plate that was smashing against the North American plate, essentially where you have the Bahamas. And then, and so essentially, there have been a lot of geological processes that have led to very complex geological formations on the islands. We could literally spend 2 weeks probably just talking about the geology and the overall evolution of the islands themselves. But the point I want to get across to you is that We have a really amazing system of tropical dry forest, lowland wet forest, and high elevation montane forest that you can find on the islands of Hispaniola, Cuba, Puerto Rico, Jamaica, and they're derived from these underlying geological processes. If we think about the overall evolution of the flora on the islands of the Greater Antilles, that's also a super complicated process, and there have been multiple ways in which the islands have been founded by species from across many different areas. I'm just going to kind of focus on the Americas here. Antonelli actually put out a nice paper in 2018 that was focused on the Amazonian region, and so he was looking at, or I should say those authors were looking at migrations into different parts of the Americas from Amazonia. And, unsurprisingly, there were quite a few linkages between the West Indies or the Caribbean region and Amazonia. So sorry about that. So as you guys can see here, I wanted to give you a couple of other examples too, and just sort of thinking about biogeographic links and what we know, or what we, sort of think we know about how the evolution of the forest in the Greater Antilles has unfolded. And so taking a look at that from, sorry about that, you guys. Taking a look at this from sort of a tropical dry forest perspective, here with the genus Melocactus. This is one of the groups that we work on in the lab. We do also see this connection with Central and South America with regards to the establishment of those species within the Greater Antilles, specifically and mostly South America. If we look at Magnolia, there was a recent paper published by Velchin et al. That showed an even more complex picture where you had dispersal out of South America, Central America, and even North America into the Greater and Lesser Antilles. Once in the Greater Antilles, with Magnolia at least, there seems to have been sort of a stepping stone approach to dispersal from one island to the next. And that is essentially how diversity was generated within Magnolia, at least in this one clade of Magnolia that's so abundant in the high elevation montane regions of especially Puerto Rico, Hispaniola, and Cuba. If we look at the family Gesneriaceae, Perret et al. In 2012 also showed some really interesting linkages between Andean South America and the West Indies. And so essentially sort of the picture that I'm trying to give everyone here is that the flora of the Greater Antilles and the Caribbean in general has been generated from a complex set of different processes and dispersal events that have spanned, Central America, South America, North America. There are examples of linkages with Africa as well, which is something that I'm not pulling in here. But so the overall evolution of these forests has been quite complicated, over the past, say, 20 million years. So what about Miconia in the Antilles? What is going on in the family Melostomataceae and within this really large clade that's so diverse in the neotropics? It's actually a really fascinating pattern that we see, given that. So there are roughly 383 species of Miconia in the Antilles. Roughly 342 of those are endemics. And I say roughly because we're still finding new things. We're still describing new species relatively often. And so these numbers are certainly subject to change. But essentially what we see in Miconia within the Antilles is that we have 4 major radiations. And those consist of, I have these color-coded here, the Antillean Sagrea clade. And it's kind of a little misleading just to say Antillean Sagrea, because there are actually 2 major clades within Antillean Sagrea that have radiated, and so have led to relatively large radiations, overall encompassing about 45 endemic species from this one clade. We have the Chenopleura clade, which consists of roughly 55 species. And this is a really interesting group that's nested within this larger clade we call Cremanium. we've got the Miconia clade of roughly 25 species that occurs within the Greater Antilles. It's essentially restricted to the Greater Antilles. And then we have this really massive clade, the Caribbean clade, of roughly 160 species. And again, these numbers are all estimates at this point, just based on what we know so far about the Miconias of the Antilles. So the Caribbean clade, of course, here being the largest and really the most widely distributed across the Antilles, given that there are some minor radiations within the Lesser Antilles as well. But in general, most of these radiations have been most successful within the Greater Antillean Islands. One other thing I should mention is that we do have isolated endemics that are sort of scattered throughout the rest of the clade of Miconia. So you can see these little terminals here in yellow. Sometimes we might have one or two species that are associated with these isolated endemics. In some cases, we have a really widespread species, like if anyone is familiar with Miconia levigata, there's actually an endemic species that's a putative sister to that species that is probably derived from levigata in one form or another, Miconia pyramidalis, that occurs within the Antilles. We also have, that I should mention, and which actually, becomes part of these overall numbers in terms of Miconia and the Antilles, lots of widespread species that are either Central or generally South American in origin. And so many of the overall numbers of species that you see in the Antilles, part of that is actually coming from these really widespread species. So you might have seen Miconia mirabilis or Dodecandra or some of those other really common species, and those are abundant in the Antilles as well. So some of these, so again, talking about some of these widespread Central and South American species, which are sometimes super straightforward in the Antilles, but not so straightforward in the continent. I just wanted to give one example of that here with Miconia racemosa. This is sort of material that from Dominican Republic. It's also in Jamaica, so it's relatively widespread in the Greater Antilles, and then of course super widespread in South America. What do we know about the biogeography in general of Miconia cincillata within the Greater Antilles? So this is from Fabian's recent paper from the book, and looking at biogeography across the Americas of Miconia cincillata. Essentially what we have in the Caribbean with these major radiations that I was talking about are linkages to Amazonian Miconia and Central and South American Miconia, specifically Andean Miconia from the northern Andes, and then sort of this widespread Andean clade. And so that's really, dealing with Macranium and the Chenopleuroclades have this Andean origin. These clades, we have some putative dates for those ranging from around 11 million to 3 million years. And so if we think about the uplift of some of the major mountain ranges in the Greater Antilles, we actually have what appears to be a correlation with some of the diversification in this group and the uplift, or the overall existence of available habitat, this montane habitat in the Greater Antilles. And so I should mention that the Caribbean clade is the oldest clade within the Greater Antilles. It is the largest, and so it seems to make a lot of sense that with mountain building within the Greater Antilles and dispersals from other parts of the range of Miconia into the islands, you would have had the origin of a diversification in the Caribbean clade. So if we look at the overall biogeography of the Caribbean clade, this is something that we're working on at the present. So this is a plastome data set that we've developed. So shout out to Lucas Bacci in the audience who has helped develop this. Essentially what we're seeing, so if we're sort of, whittling down, going from sort of a broad scale picture of Miconia. So we realized that there has been, movement from Central and South America into the Antilles. Now we're really interested in trying to determine, what has happened among the islands, right? And so essentially what we're seeing in terms of this really large Caribbean clade of about 160 species is that we're having what appears to be an origin in Cuba, and then from Cuba, and quite a lot of diversification in Cuba, likely for a number of different reasons. There are high levels of serpentine soils there, and just because of the overall complexity of the geology there, there's likely close correlation with that and diversification on the island. But from Cuba, there have been quite a number of what seem to be very clear dispersal events into Jamaica, into Hispaniola, and then from Hispaniola into Puerto Rico. But there have also been events from Cuba, dispersal events into Puerto Rico. And so there's really complicated pattern moving from one island to the other in the Greater Antilles that have led to the overall diversification patterns that we see so far across the Caribbean clade. Should also mention that from the Caribbean clade, there have been dispersals into the Lesser Antilles with Carianthus clade, for instance, a group that Darren Penny's has worked extensively on. So within the Caribbean clade, essentially, we see a Cuban origin and then movement into the other islands throughout the Caribbean. I wanted to step back a little bit to a paper that we published in 2015, where we were trying to get at the overall diversification of this smaller group. It's a subset of the Caribbean clade, one that we called the sandpaper clade, because many of the species that occur in this clade, such as Miconia lima, have these really rough upper leaf surfaces that makes them kind of fill and look like sandpaper. So in terms of looking at overall diversification among mountain regions and looking at dispersal events from one mountain region to another, we essentially see a very similar pattern as to what we see from this broad scale picture and looking at the overall Caribbean clade. And that's in this group, at least, we see this Cuban origin. And then specifically from several mountain ranges from southeastern Cuba, we see dispersal events into Jamaica and then into different mountain ranges of Hispaniola, and then from Hispaniola into Puerto Rico. So this kind of looks a little bit like the pattern that has been found, for instance, in Magnolia, where you see sort of this stepstone approach to dispersal from one major island to the next. Of course, this is sort of like a very simplistic view and a very sort of reductionist view of what's actually going on, within these mountain ranges and from one mountain range to the next. But it kind of gives us an overall sense of what might be happening at these lower levels. And so, I do want to mention as well here, and this is something that needs a ton of work still, but if you look at the Caribbean clade of Miconia across Cuba, as I was mentioning a moment ago, they occur in a wide variety of soil types. Many of those, however, do occur in serpentine soils. And even from this sort of limited phylogenetic scope from our paper in 2015, it did seem relatively clear that there had been movement from serpentine soils into clay and limestone soils. Which is quite interesting, because oftentimes we think of the adaptation to serpentine soils as being relatively restrictive, and it then being relatively difficult for plants to adapt out of that. But it seems to have happened repeatedly within Miconia, within certain groups of Miconia at least. And I should mention that the Caribbean clade of Miconia is not the only group of Miconias that occur over serpentine soils. That's actually this, adaptation lateritic soils is pretty common across Miconia. So, just sort of further narrowing our scope here, I would like to take a moment and think about the island of Hispaniola. So again, we've sort of already talked a little bit about the geologic history of the island. We oftentimes split the island of Hispaniola into two different islands, just because that's part of its geological history. So oftentimes when we're referring to the island of Hispaniola, we'll talk about the South Island, which you can see here, and then the North Island, And so essentially what we're referring to is this ancient geological formation where the South Island was at once part of one, separate landmass. And then the North Island was part of a separate landmass. And so this is the area that actually came together in the latter part of the Miocene to form what we now know as the island of Hispaniola. So across the island of Hispaniola, we have some major mountain regions. I wanted to just outline those really quick. So we have what's called the Massif de la Haute here in the lower Tiburon Peninsula of Haiti. We have the Massif de la Selle, Sierra de Bauruco range, which is here, and the southern part of Dominican Republic and southeastern part of Haiti. One thing that it's hard to tell from this map is there are actually two ranges to the Sierra de Bauruco. There's the western range and there's the eastern range, and I'll talk about that a little more in a moment. We've got the Sierra de Neiva, which is right here in sort of the southern part of the North Island, the Chain de Cajos or Sierra de Cajos. We've got the Massif du Nord, Cordillera Central, which is this really large, the largest mountain range that you see within the central part of Dominican Republic going to the northwestern part of Haiti. And then, of course, we have the Cordillera Septentrional. There are a few other sort of scattered high elevation areas out here in the east, but those tend to be less. There are melastomes there and there are Miconias there, but they tend to be less species rich than these other mountain ranges that I have outlined for you here. And so this is a schematic from Walt Judd's monograph of the Cunnipluriclade that gives a really nice sort of clear outline of these mountain ranges. So within the island of Hispaniola, it's important in terms of the diversity of Melastomataceae in the Greater Antilles. We've got about 200 species of Melastomataceae that occur across the island, so that's not an insignificant number. About 111 of those are endemic to the island, so a high level of endemism here within the family Melastomataceae. Of the 200 species, we have roughly 124 of those are Miconia, and then about 96 of those are endemic. So there's a very high level of endemism in the genus Miconia across the island of Hispaniola. With regards to overall species richness, there's high species richness in the Macranium clade, Cunnipluriclade, and especially the Sagria clade as well, and to a lesser extent, the Caribbean clade. The Caribbean clade tends to be a bit more diverse on Cuba, but there are lots of members of the Caribbean clade on Hispaniola as well. So I would like to jump into a couple of different mountain regions and just think about diversity within those smaller areas. So this is a data set that we've been working on for a while now. It's something we need to publish relatively soon. But the Massif de la Haute is one of these areas on Hispaniola that's incredibly important and incredibly interesting from the standpoint of diversification of Melastomataceae. And so if we look at this one small mountain range in southwestern Haiti on the Tiburon Peninsula, so what's called Massif de la Haute, this one mountain range, there are 63 species of Melastomataceae that occur in that one area. Of those 63 species, 43 of those are endemics, and 37 of those endemics are just Miconia. So we have a pretty good understanding of the flora of Massif de la Haute, mainly because of Eric Ekman, who did tons of work there in the early 1900s, and then Walter Judd and Dan Skeen later on, especially in the 80s when Walt Judd was working on a floristics project of biosphere reserve there. And so we have a relatively good idea of the numbers of species there and the numbers of endemics. But one thing that we can see, in terms of being interested in how that diversity has developed, the question then becomes, okay, is all of that diversity based on in situ diversifications within these specific clades that occur there, or are they just multiple separate events leading to the occupation of Massif de la Haute by multiple clades from across Miconia? So we do have, Marianias that are actually a high level of endemism of Mariania, is the highest on the island of Hispaniola that occur in Massif de la Haute. There are a couple of Henriettia species that occur there. But just focusing on Miconia, you can see a very sort of, particular pattern here. So we've got Sagria, Caribbean clade, Macranium, and Chenopleura that are outlined here. And there's a relatively large in situ diversification of Sagria within the Massif de la Haute. If you look across the Caribbean clade, there are widespread Caribbean species that occur in Massif de la Haute, and then there are localized endemics. Sometimes these are a couple of species, but most often it's just one species that occurs in Massif de la Haute. And if we look at this from a biogeographic point of view, we can see that there have been putative dispersals from Cuba as well as from other mountain ranges, especially the Massif de la Selle and the Sierra de Bauruco, which are still part of the southern island of Hispaniola, into Massif de la Haute. So essentially what we see in the mountain range of Massif de la Haute is there have been multiple dispersal events that have led to the overall diversity that we see within the Massif de la Haute itself. And there have been a lot of sort of in situ speciation events that have occurred because of those putative long distance dispersals from other mountain ranges, relatively nearby mountain ranges, involving mostly southern Cuba and then other mountain ranges of the island of Hispaniola. So if we take this down a little further and go into an even smaller area, so say for instance looking at the Sierra de Bauruco, and this is what we call the Sierra de Bauruco Oriental, because if I can just draw your attention to the map in the left-hand corner of the screen, we've got the Sierra de Bauruco Oriental. We've got this really phenomenal geological formation called the Hoyo de Pelempito, which is essentially this major depression that occurs between the Sierra de Bauruco Oriental and the Sierra de Bauruco western portion. And it essentially cuts off this mountain range, one side from the other. We think the Hoyo de Pelempito is actually, in a sense, aided diversification between these two areas, because it essentially sort of acts as an impediment to dispersal. Or potentially an impediment to dispersal, which could set up some sort of reproductive isolation scenarios. But we don't really know a lot about the Hoyo de Pelempito. We actually have a small project that's coming up next year where we're going to be doing a lot of work within the Hoyo de Pelempito to try to characterize the vegetation there. But that's kind of an aside. So getting back to the Sierra de Bauruco on the east side, one thing that's really fascinating about this area is that it's just absolutely loaded with endemic species. And to give you one example of this, just looking at the Chenopleura clade, this one large radiation within the Greater Antilles, if we just look at this, the Chenopleura clade in just one tiny area, so this is actually just one area that we work called Monteada Nueva, within the Sierra de Bauruco Oriental, we can see a wide variety of members of the Chenopleura clade. But what's interesting about this is most of the members that you see here aren't necessarily one another's closest relatives. There are exceptions to that, but in general, within these sort of confined high peaks in the Sierra de Bauruco Oriental, oftentimes what we're seeing are a really large assemblage of species within the same clade, but they're not one another's closest relatives within that clade. And so it seems that this type of diversity is likely generated from dispersal events from different mountain ranges into this smaller area, similar to kind of what we're seeing in Massif de la Haute. I should mention that there are Chenopleura clade members here. There are also many other members of the Caribbean clade, Sagria, Macranium. And so when you walk into one of these forests, you see just an incredible diversity of Miconia within these forests that have been generated from numerous different clades. So if we're thinking about speciation within the mountains of Hispaniola specifically, There seem to be a number of different patterns that pop out. And one of those that I would like to discuss here in Massif de la Haute is this potential niche partitioning. And so within some areas of specific mountain ranges, if you work your way from low elevation to high elevation parts of the mountains, you will see that these close relatives appear to sort of distributionally, they settle out along an elevational gradient. And so essentially the idea here is that probably there is a bit of niche partitioning where you have, for instance, in this case, I wanted to share with you Miconia subcompressa and very close relatives. So Miconia subcompressa on the lower left-hand corner of the screen, this is a taxon that generally occurs in what we call low elevation rock bois. So this is like dogtooth limestone. And it might occur in elevations from about 800 to maybe in upwards of 1100 meters, something like that. And then as you start to work your way up the side of the mountain, in this case, what would be Mont Faumont, you start picking up this entity, which we've called Miconia subcompressa subspecies plumeri, because there are characters that kind of grade into typical subcompressa. And so you'll start picking this up around 1100, 1200, and it might go up to, 13, 1400, something like that, sometimes even a little bit higher. Then as you work your way up into the higher part of these, what at this point are cloud forests, you start to pick up Miconia sinotricha. And so in one of Walt's papers in 2015, we actually show that Miconia sinotricha is nested within subcompressa. And so this kind of appears to be this really interesting way that some of these taxa are diversifying just by occupying different niches along an elevational gradient. This doesn't seem to be the most common pattern that we see in Miconias, specifically on the island of Hispaniola, but it is something that appears to be happening in specific mountain ranges in specific clades. And so this is just one example of that. From the Massif de la Haute. What seems to really be the common pattern are long distance dispersal. And so what I mean by that is you oftentimes have one species that occurs in a very specific mountain range, and then its sister species that occurs in a totally different mountain range. And so this is one example of what I'm referring to here. We've got Miconia campanensis that's known from a few sites in the Cordillera Central. And then we have Miconia howardiana, which is essentially restricted to eastern Sierra de Bauruco, that area of high endemism that I was talking about just a moment ago. And so, essentially what we appear to have in many of these situations are kind of like, Sky Island types of situations. So you can imagine a dispersal event from a founding, leading to a founding population from the Cordillera Central into the eastern Sierra de Bauruco. And then, of course, with different environmental constraints and selective pressures, you have speciation and differentiation from one species to another. I know that these species look a lot alike. They're actually pretty good morphological characters you can use to separate them. So, don't just take it at face value. They actually probably are, really good species. But we see this quite often, right? Where we have patterns. We still have a lot of work to do to differentiate or to actually look at the directionality of many of these patterns or many of these dispersal events that we're seeing. But this is sort of a repeated pattern. So I wanted to give you a couple of other examples of this. So we're actually in the process of describing this species from the Sierra de Neiva. This was just recently discovered a few years ago by Teodoro Clase, who you can see here standing beside this relatively large Miconia. This occurs in Sierra de Neiva in just a couple of populations. And then we have its closest relatives are Miconia bardiana and Miconia impressonervis, both from the Cordillera Central. Impressonervis, the yellow dot you can see here is kind of on the southern slopes of the Cordillera Central, and bardiana is in serpentine soils on the northern slopes of the Cordillera Central. So there are a few things happening here which seem to be quite interesting. It actually appears that Bardiana is probably most closely related to this new species from the Sierra de Neiva. But so this does appear to be a pretty clear situation where we've had a dispersal event from one mountain range to another that sets up reproductive isolation and speciation. But then within the Cordillera Central itself, just because we're in the same mountain range doesn't mean that, these species are actually sympatric. They actually occur in really different types of habitats on different soil types. And so even within the same mountain range, oftentimes we're not seeing very much symmetry. And so between different major peaks within the Cordillera Central, we're also getting sort of these short range dispersal events, apparently, in some situations that are likely leading to speciation events. And so this is a really interesting pattern that we see from the Cordillera Central into the Sierra de Neiva. Here's another one that's quite fascinating. So we have Miconia pañolensis that was just unearthed, back in 2013. Joel Timian, who works for the Audubon Society of Haiti, was doing some really interesting helicopter work with Blair Hedges, and they ended up helicoptering into some parts of the mountain range of Massif de la Roche that people had not been to. And of course, they ran across new species. And so this was a species that was uncovered during that work that he was doing there. And then just recently, we've found another new species from the Cordillera Central from a number of different locations. There are a couple of sterile specimens that I've seen, and then I've collected this fertile in one location as well. We actually have this in the phylogeny now, and it was a bit of surprise to see the relationships in the phylogeny, and that Añolensis was sister to this new species from the Cordillera Central. And so if we're inferring, how speciation was actually set up in this situation, it does appear, very likely that there was a long distance dispersal event from one place to the other. We don't really know the directionality of this dispersal event, but one thing that we're planning to do once we have a bit more comprehensive sampling across the Caribbean clade is to sort of go back and revisit movements among mountain ranges within the islands themselves, so we can really start to develop much more comprehensive ideas about how many of these biogeographic patterns were set up. So one last one that I'll give you in terms of looking at sister species and different parts of mountain ranges. So this is kind of getting back to what I mentioned earlier regarding the Olla de Pelempito, this really interesting geological depression between the eastern Sierra de Bauruco and the western side and Massif de la Selle. So we have Miconia turbinata, which occurs in Massif de la Selle and Sierra de Bauruco. And then we have a species actually we just described in 2018, Miconia classei, that occurs on just one peak in Sierra de Bauruco Oriental. And so so this is one of those situations where we have, instead of having a contiguous mountain range from Massif de la Selle all the way over to the east, it's actually broken up by the Olla de Pelempito. And so this presumably would, in some degree impede or cause reproductive isolation between populations of, say, turbinata and classei that occur here in the east, therefore setting up, this speciation. So this is a really interesting pattern that we do see not only in these two species, but in quite a number of other species of Miconia. In the Sierra de Bauruco, Massif de la Selle, and Sierra de Bauruco Oriental. Okay, what about sympatric species? I've been talking about, essentially, speciation along elevational gradient, speciation via long distance or short distance dispersal. But getting back to Massif de la Roche, it's this really special area where we have many species of endemic Miconia. And we actually do have a couple of situations where there seem to be in situ radiations, and the taxa do not always sort out along an elevational gradient within these areas. And two examples of that I wanted to give you are Miconia section Sagrioides. So this is what we call the Macranium clade, and then Miconia, the Brevissima clade. So this is one of those in situ radiations of Sagria. And what we see here in some of these low elevation, especially within the low elevation occurrences of Macranium, we see putative close relatives that are growing relatively close to one another, sometimes intermixed. And so in this case, there's definitely something else that's going on. It does look like we potentially could have a certain type of sympatric speciation going on within these areas. Many of these taxa that occur close together are growing on this dogtooth limestone that you can see here, what we call Racbois in Haiti. And they might grow in some slightly different microenvironments in some cases, but it's super interesting because you see many of these taxa growing close to one another and sometimes intermixed altogether. And so there definitely is a situation within Massif de la Hoda especially, and this does apply to some taxa on Cuba as well, especially in the Ecanati clade, where we have a putative sympatric speciation going on. And so it's quite a different pattern than what we see most typically across the mountain regions of especially Hispaniola. So to sort of wrap up here, I'm a little long-winded this morning. So in looking at the broad scale patterns across the Greater Antilles, we have a lot of the diversification within these groups likely driven by long distance dispersal events to these different mountain ranges or different peaks within the same mountain range that very easily sets up speciation via reproductive isolation. We do have what appears to be niche partitioning and specialization within given mountain ranges. And then there are certain groups that appear to be, undergoing radiations sort of all within the same type of habitat, really around the same elevation. So what appear to be some sympatric speciation events going on. There are tons of unanswered questions. I've sort of just truly skimmed the surface during this talk. So what do we know about pollinators? We don't know hardly anything about pollinators of this group in the Greater Antilles. We do know that insects are doing some of it, birds are doing some of it, bats are doing some pollination. There's a record of Miconia synthenesi from Puerto Rico of the Quinoplura clade, which is pollinated by bats. I talk about dispersal quite a lot, but who's doing all the dispersal, right? It would be really nice to have some detailed studies looking at, different types of birds that potentially could be helping to explain some of the patterns that we see. What about hybridization and polyploidy, so whole genome duplication? That's something that is totally unexplored really at this point. We have one putative taxon derived from hybridization, is the Miconia markgraffi. It's a putative hybrid between Miconia subcompressa and ferruginea within the Quinoplura clade, but that it hasn't been verified. It's just sort of looking at morphology. We're actually looking into this scenario at the moment in my lab. But there's a couple of other situations and a few other different clades where we might have some hybridization going on, which could potentially lead to cryptic species and further diversification. And so that's something we would also want to do in looking at these sympatric radiations. What about insular level phylogeography across the Greater Antilles? This is something that has not been done yet, but which would be super enlightening, taking some of these widespread endemic species, either single island endemics or multi-island endemics, and doing some very fine-scale phylogeographic work to look at patterns of dispersal. So I'm going to finish up there, just to acknowledge a number of people that without their help, everything I showed you today would not have been possible. So, and then of course funding for some of this work, NSF, and of course, Florida Museum. I'd be happy to take any questions anyone might have.
Okay, very nice, very nice. So we have some time here, and if you want to ask any question, just please unmute yourself. And yeah, so who is one question? Who is a black one white there in the acknowledgement? Is that Ekman?
That's Ekman. Yeah, that's Ekman. I had to put him in there. For those of you who do not know who Eric Ekman was, he was a crazy Swedish botanist. I say crazy, he was just like, he ate, drink and slept plants. That's all he did. And so he worked on, well, actually he for his dissertation, he worked on Vernonia, and he did a lot of work in Southern South America, specifically Argentina. But then he ended up getting hired by Ignatius Urban, and worked first of all, on the island of Cuba, and collected, I don't know, around 19,000 numbers, many of those with 3 or 4 or 5 duplicates apiece. And then he went to the island of Hispaniola and collected another, 19,000 or so numbers before dying at the age of, I believe he was 42 when he died from malaria in Santiago, Dominican Republic. But he was so this is a photo of him coming down off of from one peak in Massif de la Haute, with all of his amazing collections there. And honestly, some of his herbarium collections are the most beautiful collections I've ever seen. They're just phenomenal, absolutely phenomenal. We've actually even been able to, melanostomes are really problematic for extracting DNA. We've actually been able to extract DNA from some of his specimens collected from Massif de la Haute. It's, because they were so well preserved. So without, all of Ekman's work across Hispaniola, we would have no idea that some of these things occur in specific mountain ranges where we haven't been able to get back and collect them. So anyway, that's Eric Ekman.
It's amazing. Now I know where Mariana Ekmani comes from.
Yes, that's right.
I've been wondering. Thank you so much, Lucas, for the talk. That was really cool. Like a wonderful overview of different processes that are going on. And I have several questions, and I'll start with maybe one that could be slightly ignorant. I've been seeing this pattern not only in your talk, but also in Mariani and in other talks, that it seems that melastomes colonized the Antilles through sort of the northeast and never through the, northwest and never through the Lesser Antilles. Is this because the Lesser Antilles are of more recent origin? Because geographically, it would be so much easier to go through the Lesser Antilles and then spread north. And this doesn't seem to have happened at all, which I think is very surprising.
That's really, really fascinating. It's really fascinating. And the other thing about that pattern is you see that pattern in so many other groups as well. You actually see it in cacti as well. You see that's a pretty common pattern in cacti, actually. And so I think presumably part of that could be because the younger age of the Lesser Antilles and just the lack of as much available habitat in the Lesser Antilles for that type of pattern to have occurred. One thing I didn't talk about, which is another potential route, but it doesn't really seem to apply to melastomes, right? Because they're too young in terms of what at least we think we know about when they arrived in the islands. But Garlandia, many of you have probably heard of Garlandia, the Garlandia hypothesis, but this was a putative land bridge that would have arisen in the Oligocene. So we're talking 34 million years ago, something like that. So it's really totally inapplicable to this situation, but that actually would have cut across sort of just to the west of the Lesser Antilles in the way we know them now. But yeah, I think it's probably a combination of the overall lack of large land masses for species to sort of stepstone across, and lack of habitat within the Lesser Antilles for them to do that the way they would need to have made it to the Greater Antilles. Yeah.
Cool.
Fabian.
Yeah. So what one exception there might be, but we don't have enough data yet, is some of those Sagria. They might have come from the Amazon through the Lesser Antilles. And then for Henriettae, it's kind of weird because it seems that they are close. The Antillean ones are more closely related to things in Amazonia or the northern part of the Andes, but they sort of have skipped Central America, maybe. I mean, there are species of 110 Central America, but they're not the most closely related ones to the Antillean. But yeah, that's still, yeah. And then of course there are 2 species of Rexia in Cuba that came from the north.
Yeah. Yeah, and yeah, Rexia cubensis is also on Hispaniola and on Puerto Rico as well. So yeah, there has been sort of that Magnolia, North American into the Antillean pattern as well. Yeah, I guess so that makes sense in terms of Sagrea, perhaps, given that a lot of those occur at lower elevations, not all of them, but so maybe that could help explain why you might see a pattern, potentially going through the Lesser Antilles for something like Sagrea. Yeah.
It was also really cool how you showed sort of different processes of speciation that could be happening, like niche differentiation or long distance dispersal, allopatric speciation, and so on. And I was wondering how, like, it sounds like the ideal system, island systems, I don't know, to study that. But then how sure are you that you're not missing populations in between? Could you even, I don't know how destroyed the natural habitats on these islands are, whether you could be missing sort of extinct populations or not. I have no idea, so I'm neutrally asking here.
I think that's a fantastic question. That's a fantastic question because we could very well be missing. So actually what I just showed you with regards to one of those new species and its relative in Massif de la Haute, so Cordillera Central, Massif de la Haute, I mean, that new species, that's something that we just actually came across in 2019. So without having uncovered that, we would be totally missing that pattern altogether, right? And we would actually see a pattern that would look more like a Cuba Massif de la Haute pattern. And so
sorry, this fire alarm is going off in the building. I need to evacuate, but I'm gonna leave this on.
Okay.
So keep it on in and whatever.
Okay, sounds good. Yeah, so really great question. And I think that's, there are a couple of different things that I could say about that. Yeah, definitely we're missing populations that would really help us understand these patterns a bit better, and that might actually fill in some of the gaps. But there has been quite a bit of destruction in a lot of these high elevation montane forests. And so some of these areas presumably, that could have housed species that are, or populations of species already known, we could be missing the boat on some of these patterns just because we don't have access to either, other populations or, species that are just totally unknown. Or, we're still actually uncovering quite a few new species when we make it into areas that haven't been super well explored. Yeah. And so, we're slowly filling in the gaps here and there, but there's still so much work that needs to be done.
Yeah, very related to that, Darren asked in the chat how complete the sampling from the Lesser Antilles is and whether there could be some changes to the story.
Oh, I think absolutely. We're missing sampling from the Lesser Antilles. We need better sampling from the Lesser Antilles. So I think that could affect the story a little bit. Yeah. I do think that in general, say for instance, within the Caribbean clade, I think it seems to be pretty straightforward that there was an out of Greater Antilles dispersal into the Lesser Antilles. But perhaps, for some of these other groups like Sagrea, for instance, if we did have a more comprehensive sampling across the Lesser Antilles, it could potentially give us a slightly different story. Yeah.
Are there any other questions?
I have one.
Go ahead.
Great talk, amazing work.
Oh, thanks, Lucas.
My question is regarding the distribution of these species inside the islands. For example, in Miconia, even when you have a really widespread species inside one island, it's really uncommon that this species colonize other islands, right?
Yeah.
So it always tricks me because when you think about dispersion, those islands are super close to each other, right? So why do you think that this is a pattern?
Oh, that's a great question. It's a really tough question. I brought my slides back to Miconia hispidula because that's one on Hispaniola that's essentially everywhere. and it occurs in relatively low elevation. So we're talking, 500 meters, well, let's say 400 up to like 800 meters, something like that. And there are a lot of really great habitat for it across the island of Hispaniola. And so it's pretty much everywhere across the island. But it's restricted to Hispaniola. Why it hasn't made it to Cuba or Jamaica or Puerto Rico is honestly beyond me. Perhaps it has something to do with a specific dispersal agent. Maybe it's really good at dispersing locally from sort of one, low elevation part of the mountain range to another, but maybe it doesn't have a long distance dispersal agent. And it does form these really large fruit, which you might think could be eaten and dispersed by something like, these gulping birds like thrashers, which don't tend to have really large territories, I guess you could say. So perhaps, it's more tied to the dispersal agent or something like that. That's a really great question, and I don't have a good answer for you. Maybe it could also be tied to just the overall age of the species and its populations. Maybe it hasn't had time to make it to other islands yet. But yeah, it's a great question. And I don't have a good answer.
We have a related question about age in the chat from Kalle, who asked or who commented that your sister species that you showed from separate mountains looked surprisingly different morphologically. And whether you have any idea of when they separated?
Yeah, those are sort of really fine details that we're still working on, but in general, they seem to be relatively recent, actually quite recent within the Pleistocene, or, within that time frame. And so they seem to be very young divergences from one another. But it is the case, like this one new species from Sierra de Neiva. I was actually super surprised when it fell out in the phylogeny where it did, because I had, phonetically, it looks like something that's in another part of that same clade. But, I think maybe that's just one other really good example of homoplasia in Miconia. Homoplasia is super rampant in Miconia. And so, so it is, sometimes these close relatives are surprisingly morphologically divergent.
How important were the sort of glaciation times in the Antilles? I have no idea. I mean, did the higher peaks ever, like climatic fluctuations during the Pleistocene?
So there's some really fascinating literature on putative glaciation in the Cordillera Central. And so there is the possibilities. There's some really interesting geological formations and actually some high elevation savannas in the Cordillera Central. And there is potential evidence that those areas might have formed through glaciation. And so it does seem that glaciation during the Pleistocene could have, at least in one way or another, augmented, the distributions and sort of speciation patterns that we see in groups like, montane Miconias. So yeah, it's so the highest peak in the Antilles is Pico Duarte at about 3,300 meters. So it's not like, the Andes, but
it's pretty high still.
But it's pretty high. It's pretty high for an insular system, right?
Yeah.
That's precisely, within the Cordillera Central, that would have been one of the areas that could have potentially been affected by glaciation.
Cool. Do we have other questions? We're slightly over time.
This is Doug Stone. Can you hear me?
Hi, Doug.
Great talk, Lucas. Thank you. Sympatric speciation. So we know from both theoretical and empirical studies that sympatric speciation is a thing. In other words, if selection is strong enough, then these lineages can be maintained and continue to diverge in sympatry. I'm just wondering if from the cases that you're aware of right now, are there any clues in morphology about what might be driving?
Yeah, that's a really, really good question. Really good question. So the two examples that I gave you, within the Sagria clade and then within the Macranium clade, morphologically, they're those groups are really similar. It's kind of like iterations of the same common theme. So in Micranium, you have these axillary inflorescences with really, really tiny flowers. We have no idea what's pollinating those. I know Dan Skeen has seen flies and things like that go to those flowers. But they're all growing, and many of those are growing in sympatry right there in Massif de la Haute. And it's really unclear what might be driving or what might be maintaining those species boundaries. One thing that we don't have a clear idea of is ploidy within those groups. So we don't know if perhaps some of the sympatric speciation, putative sympatric speciation we're seeing could be, sort of a consequence of, sort of ancient hybridization and polyploidy or something like that. That's one area where we really need some nice data sets to see if that could actually be sort of one of the mechanisms by which they are speciating within those groups. But at this point, I don't have any great hypotheses as to how they're maintaining species boundaries.
Great, thank you.
Thanks, Doug.
Hi, this is Julie Denslow. I'm wondering whether or not there may be mechanisms setting up reproductive isolation sort of instantaneously or very quickly in some of these populations so that you could get divergence even though you don't have niche differences.
Oh, sure. Yeah. I could envision that you could get something like that. Just in perhaps contextually based on which clade, you were looking at. But I think that there definitely is the potential for that, even without differentiation of niches and, without even invoking something like polyploidy. I think that that's definitely a viable hypothesis.
Cool. I've always been struck by the ecological convergence of melanosomes. In both their pollinators and their habitats and their dispersal agents. So
absolutely. And that's one thing that's fascinating. Like, in the Quinopluriclade that I showed you, that one example from Sierra de Bauruco, many of those taxa grow side by side, but they're from different parts of the Quinopluriclade, so they're not one another's closest relatives. And so they're essentially occupying very similar niches, but they're doing things in a way where there doesn't seem to be, really much hybridization at all. And so presumably there are some pretty strong barriers to, reproductive isolated barriers. Yeah.
Cool. Thanks. Very nice talk.
Thank you. So Agnes, just, this is one place where we need some pollination biology. It would be so
well,
nice.
I've already asked you when you're going next. So
yes,
I'm toying these ideas.
Yes,
but related to Julie's question, I've now just wondered, do you think that the symmetry that we see today is actually where the species evolved? Or do you think it is sort of secondary symmetry, or could be? Do you have any sort of data set that is detailed enough where you could look into this?
I think that's a super complex question.
I know.
Yeah, super complex question. That kind of gets back to this idea of putting together some phylogeographic data sets to look at the patterns across, for instance, Hispaniola, and actually look to see if we could get at your question, right? And look at the putative origin of some of these taxa and whether or not, the areas where we see those in symmetry, are those just secondary dispersals into those areas or is something else going on? Yeah.
Cool, thank you. I see Fabian has not burned, but is back.
That's good. Fabian's he's safe.
Our engineering team is not the smartest sometimes, and they were doing something else and tripped the alarm.
Oh.
Okay,
but we still have to wait for them to give us a clear to come back in. So I don't know if you answered a couple questions that were here in the chat. Okay.
Yeah.
So again, we'll see you. Are we close to done? Are we done?
I think so. I was just going to ask, were there any other questions? Any last burning question?
No pun intended. So great. We'll see you guys next year, and thank you very much. And look for the recording of this soon. And again, volunteer to give a talk. We're here for you guys. And see you soon.